Good morning, everybody. Welcome to another edition of the Lab 207 webcast. My name is Mr. Kite. I'll be hanging out with you today. Today we have got our second of two videos about cell signaling. We're going to talk about the actual process of signaling. Now, before we get into this, I'm not going to lie to you. Cell signaling is not my forte, so I might use more words than I normally would to say the same thing, but I still think we can get through it all right. So as always, let's get our objectives. These are the things I need you to be able to do by the time the video is over. First thing, trace the path of a cell signal from reception to response. Second thing, compare and contrast transmembrane receptors. Third, recognize the importance of protein phosphorylation, dephosphorylation, and unrelated second messengers. And third, provide examples of response fine-tuning. Got a lot of stuff to cover, so let's go ahead and get going. First, a brief overview to get us kind of in the right frame of mind for this discussion. If we are talking about cell signaling, there are three steps that we're going to follow. The first one is the reception of the signal. Second is going to be transduction. That's where the signal is actually communicated through the cell. And finally, the response that is enacted by the cell. Let's talk about each one of those in a little bit more depth, starting with reception. Now, a term that you need to know is ligand. A ligand is any molecule that binds specifically to a receptor of some sort. Generally, they're going to be proteins. So in this case, you have got a specific signal molecule that is known as the ligand. That signal molecule is going to bind to a specific receptor on the membrane of the cell. Now, when that ligand binds, it causes some sort of shape change in the receptor molecule. I'm not going to get specific about what the shape change is, but just know that the binding of this molecule causes this thing to change shape. It changes from being inactive to active. Whatever shape it changes into, that is its active shape. So step one of the cell signaling process is the signal molecule, the ligand, binds to the receptor, and the receptor has a corresponding change in shape. Now, before we go into the other steps, there are three major categories of receptors that you need to be aware of in the cell membrane. All of them are transmembrane proteins, which means that they go all the way across the membrane. And, well, let's just talk about them in depth. So first one is a G protein coupled receptor. There's a lot of words. Thing to remember is this is a receptor that is turned on and off via GTP. It is called G protein coupled because you have got a protein that is coupled to GTP. GTP is just a cousin of ATP. ATP's got adenosine, GTP's got guanine. They're essentially the same molecules, just ATP is the more famous sibling. So in this receptor, essentially what you got is you've got this coupled protein. He's got GTP hanging off of him, or GDP. When the signal binds to our receptor, the receptor is going to change shape. When this receptor changes shape, it causes this little protein right here to take GDP and convert it to GTP, or the other way around. Once that conversion happens, a phosphate is stuck onto this guy. He changes shape and activates this one, who changes shape and activates this one, who causes some change within the cell. It could be to activate enzymes or something like that. Just know that this sort of protein receptor is hooked to a protein that converts GTP to GDP or GDP to GTP. In that process, a phosphate is stuck to the receptor, which causes a shape change and sends the signal through the cell. The second receptor that you need to know is a receptor tyrosine kinase. The thing that you need to know about these guys is that they multiply the effects. Now, here's essentially the pieces of a receptor tyrosine kinase. Before this thing gets activated, these two pieces of the signal molecule, or of the receptor, are actually separated. So split this guy in half, put half of him over here, half of him over there, and that is his inactive monomer form. When our signal proteins, our ligands, bind to him, the two halves come together to form a dimer. Okay, so that is two monomers hooked together, makes a dimer. When the dimer hooks up, he is activated, and this is a big old phosphorylating machine. You get a bunch of ATP that turns to ADP in the process, hooking a bunch of phosphates on here. This guy can hook like 6 or 8 phosphates on him. Each one of those phosphates can be used to set off a cellular response. So it's possible that using your tyrosine kinase, one signal molecule could set off multiple pathways within the same cell. Your final cell receptor to know is an ion channel receptor. This is like a gatekeeper because when the ligand binds to him, his gate flies open. Now, we talked a lot about setting up ion gradients in previous videos. This would utilize those ion gradients to get some work done. So in the normal unsignaled state, the gate is closed, the channel is closed, our ligand binds, the gate flies open, and because of diffusion, all of those ions, they rush inside of the cell, which causes some sort of cellular response. Then when the ligand goes away, the gate closes and things reset. All right, next step of our cell signaling pathway is transduction. Just want you to think of this as a series of dominoes falling over on a table. When our signal binds to our receptor protein, it changes shape. That change in shape sets off a cascade. So this change in shape might activate this protein or change him from inactive to active. The change of this one to active might activate your second protein, which activates your third protein, which activates your fourth protein. You can think of this like that cheesy game telephone where somebody comes up with an original message, whispers it to the next person, and then they pass it all the way around the circle. Now, hopefully, unlike the game, this actually sends an accurate message rather than the inaccurate message that goes around in telephone. But know that the shape change of the receptor sets off a cascade where the message is passed through a bunch of different proteins before it gets to its final destination. Now, a lot of those proteins, as they receive the signal, they are getting phosphorylated or dephosphorylated. Two terms that you need to know right here. I'm just going to underline them real quick. A protein kinase and a protein phosphatase. These have got opposite effects. A protein kinase adds a phosphate to a molecule, which in most cases would change that molecule from being inactive to being active. A protein phosphatase does the opposite. He strips phosphates off of a molecule. So in most transduction pathways, the protein kinase will turn the inactive protein into an active protein by adding a phosphate. And then when we're resetting the series to do it again, the protein phosphatase will go through, knock off those proteins and change our active, or knock off the phosphates, changing our active protein into an inactive protein. In this idea of signal transduction, we have got second messengers. Now, most of the signal molecules that we are talking about aren't water soluble. They are big proteins, are relay molecules that pass a message along. They're big proteins, so they have to move kind of slowly. They got to talk to each other. They have to connect with each other. They have to be phosphorylated, dephosphorylated. So they don't move very quickly. To help them out, your body has got second messengers. Now, on a further slide, I think I've got, actually maybe not, let me write them in here. There are 2 second messengers that I want you to be aware of. There are actually 3 of them. IP3 is one of them, calcium ion is another, and CAMP is a third. Each of these second messengers are water-soluble molecules that can diffuse through the plasma membrane, and they are able to move through the cell much more quickly. So what a second messenger does is if a receptor is bound to our receptor molecule and it sets off a transduction cascade, this cascade might release a whole bunch of these small molecules that can diffuse through the cell very quickly and have a quick and amplified effect because they can spread out all over the place. One example of this would be calcium. Our cells, a lot of our cells react to the concentration of calcium in the cytoplasm. Generally, there's a lot of calcium outside the cells, but there can't be a response until there's calcium inside the cell. So what this would look like is as our signal goes along, it would open up an ion channel, which would cause a bunch of calcium to rush into the cell. That second messenger, the concentration of calcium going up inside the cell, would set off a cellular response. So second messengers, just know that they move very quickly and they diffuse through the cytoplasm very quickly, causing an amplified response. Final piece of this mess is going to be our actual response. Now, obviously there is a myriad of responses that can result from our transduction pathway. Just know that they fall into two categories. You can either have regulation of transcription, which means that in terms of DNA, we are either going to use that DNA segment, that gene, to produce more proteins or fewer proteins. That would be regulation of transcription. Or there might be some cytoplasmic activity that's being set off. But either way, we are following our path from our ligand binding to our receptor, setting off some sort of transduction pathway. That transduction pathway might cause second messengers to diffuse in the cell and bind with some other relay molecules. Eventually, that whole transduction pathway is going to cause an effect. That effect might be causing the DNA to be transcribed more, giving more proteins, or it might be something out here in the cytoplasm. Now, I know we're starting to get a little bit long, so let's go ahead and finish up right here. Our signaling, cell signaling, can be fine-tuned in a bunch of different ways because obviously these are highly specific reactions that are happening, so your body's got to have some control over them. A couple of things that can happen. You can have signal amplification, whereby one signal molecule could turn on a whole bunch of molecules in the cell. So you could have one ligand molecule that binds to a receptor. That receptor could cause a change in two different relay molecules. Those relay molecules could each affect two more. And you could see how this very quickly could have an exponential effect where your signal goes from being very small, one molecule, to a whole cascade flying through the cell. So you can amplify a signal. Specificity of signaling leads to coordinated response. All I mean by that is that since these signal molecules are specific to the cells, they can fly past a bunch of cells until they get to the right receptor. So that's one of your ways that the body makes sure that things that don't need to be signaled aren't getting signaled. And finally, you got to be able to shut down this response, whatever's happening. Ways that the response is shut down generally are going to be those phosphatases, knocking the phosphates off, which would turn your active protein into an inactive protein. It might look like pumping second messengers back out of the cell. Either way, it's some sort of reset so that your cell is able to respond to the next signal that comes along. So I know that was a lot. I hope I didn't run long. Even if I did, I hope it was helpful to you. Thank you for joining us on the Lab 207 webcast. Hopefully we'll see you again. Have a good day.